Electron transport chain is the first half of the oxidative phosphorylation process. It involves transport of electron through a series of protein complexes, which releases energy that will be used to pump proton against their concentration gradient. Electron transport chain takes place along the inner membrane of the mitochondrion, or also known as the cristae. Inner membrane envelops the matrix of mitochondrion, while the space in between inner membrane and the outer membrane is called intermembrane space. The matrix of the mitochondrion has lower concentration of proton in comparison to intermembrane space, which has higher concentration of proton. Because of the concentration difference, this is why to move proton from matrix into the intermembrane space, the process would require energy or active transport. This is where the electron transport chain comes into play, which is to provide energy to pump proton against their concentration gradient. The electron used in electron transport chain came from oxidation of NADH as well as FADH2. Oxidation of NADH will occur at complex 1, which releases electron and then transfer the electron to complex 1. This is where the energy from the electron is used by complex 1 to pump proton from matrix into mitochondrion against their concentration gradient. Next, a mobile electron carrier molecule will transfer electron from complex 1 to complex 3. This molecule is called ubiquinone or coenzyme Q. Upon receiving electron from coenzyme Q, complex 3 will now have the energy to pump proton against their concentration gradient from the matrix of mitochondrion into the intermembrane space. Next, another mobile electron carrier molecule called cytochrome C will transfer electron from complex 3 to complex 4. Complex 4 will also uses the energy from the electron to pump proton against their concentration gradient. Finally, the electron from complex 4 will be used to reduce oxygen molecules into water. Since oxygen is the last molecule that will receive the electron, this is why in aerobic respiration, oxygen will act as the final electron acceptor. The process of electron transport chain will repeat for each of the NADH molecule produced during glycolysis, link reaction, as well as Krebs cycle. Next, electron transport chain will also involve oxidation of FADH2. However, unlike NADH, oxidation of FADH2 will take place at complex 2. And complex 2 does not pump proton across the membrane. The electron is then passed to complex 3 by coenzyme Q. Next, from complex 3 to complex 4 by cytochrome C. As we can see, both complex 3 and complex 4 will pump proton against concentration gradient once they receive the electron. Finally, the electron will also be passed to oxygen molecule to form water. Then the process repeats for each of the FADH2 produced during Krebs cycle. Overall, we can see that oxidation of FADH2 will only involve flow of electron from complex 2 to complex 3 and then finally to complex 4 and pump proton twice. In comparison to oxidation of NADH, which involve complex 1, complex 3 and complex 4 and pumping of proton 3 times. In terms of product, electron transport chain will create potential energy stored in the form of proton gradient called proton motive force. So this energy will be used in the next process, which is chemiosmosis.